Swayam Prabha. Digital India. Educated India.
Welcome back to the second lecture of the 9th week. So, last class if you remember, we talked about how the lenses form. So, I told you that there are two unique events which happens in some of the cells. I talked about the RBCs, I talked about the lens. So, in the RBC, on the contrary, so I am just going side by side because later we will talk about the RBCs, I will not discuss this topic. Okay. In the case of RBCs, what happens is, so here, so let me just put it down. So, this is, we are heading for lecture 2, week 9. So, W9 L2. Okay. So, now in the case of RBCs, what is happening is, out here where you see the expression of alpha crystalline protein in the case of lens. In case of RBC, this is replaced by hemoglobin. There is huge amount of formation of hemoglobin, which are oxygen carrying protein or metalloprotein, which has a iron sitting in the center of a porphyrin ring. Porphyrin is the same ring, very similar ring rather, pardon me, very similar ring which is used by the plants, the chlorophyll molecules. A porphyrin ring with a magnesium in the center makes chlorophyll. A porphyrin ring with the iron in the center having colored red because of the iron present there at a +2 state of iron in the ferrous state. So, that forms the hemoglobin, whereas on the contrary, in the case of magnesium, that offers it a green color, which is the chlorophyll. So, nature has, for some very interesting reason, has picked up these metalloproteins in a very interesting way. Definitely when you look at hemoglobin and chlorophyll, you feel like that. Okay. So, coming back where I was. So, in the case of red blood cells, the last major protein which is synthesized by these red blood cells are hemoglobin, whereas in the case of lens, the last protein, the major protein which is synthesized is alpha crystalline, right. So, now after this what happens? There is a sharp spike of a protein called, or enzyme rather, called 15-lipoxygenase. What lipoxygenase does? As the name indicates, if you break down the name, it is an oxygenase. It basically oxidizes the lipid. If something oxidizes lipid, that means when you talk in biology or biochemistry, anything which it is almost damaging the lipid. Now, if you look at a cell without, so this is our cell, okay. It has a membrane and this membrane is made up of nothing but lipids, lipid bilayer, right. And within lipid bilayer, you have a series of proteins which are sitting there, right. So, now, so this is your lipid bilayer, these are the membrane proteins. Which are embedded in these structures. Okay. So, now if there is a sharp rise of lipoxygenase, in this case 15-lipoxygenase, what this will do? It will immediately try to oxidize these lipids, and if it oxidizes these lipids, what it will do? It will create perforations, it will create gaps, and if it create gaps, then this cell will die out, because then the extracellular fluid will come inside, gush in, and it will do all sorts of damage to these cells. And of course, this depends how much and for how long the 15 lipoxygenase is expressed. That means if y axis is showing the quantity, and x axis is showing the time, then the important part is how long. Is it this short of a window, or this is something like this, or this is something like this, how much. It is interesting to note that 15 lipoxygenase expression is like a sharp peak. Almost the peak has almost, it has no slope, zoop like this. But that zoop is good enough, so go back, to create perforations in these structures, which is the nucleus, mitochondria, Golgi body, endoplasmic reticulum. These are all membranous structure, and a little bit of a damage on the main membrane too. But that is very negligible because it peaks up here like this, and in the surrounding vicinity diffuses in a way it damages these organelles. And once it damages, so the way it damages it, it creates a pore like this on the membrane. So something like out here, it will create a pore like this, and as soon as it creates pore, the milieu, the internal to external membrane potentials of each one of those organelles. Mind it, each one of these organelles have their own potential window to stay, right. Mitochondria has a different potential window, endoplasmic reticulum has different, nucleus has different, likewise. So as soon as these pores are created, these structures are directly exposed to the different proteases and nucleases, and these structures are now dissolved out mostly, or they almost become rudimentary. And once these are dissolved out, what you are left behind is a large chunk of this wonderful protein out there, which was expressed in a huge amount in the case of lens. And that protein, which I am now shading in blue, is alpha crystalline. So now you have a cell. I have to show you have a cell now, which is something like this. Lot of alpha crystalline present in a particular way. They are all self-assembly of alpha crystalline like this, which is okay, and you have a cell membrane like this. This is an individual lens cell, and that individual lens cells are left with some of those membrane proteins out here, which helps it to maintain its osmotic balance, and the lipid bilayer, which suffers a very least damage because of the time window of lipoxygenase activity, where lipoxygenase is very busy in chewing out the organelles instead of damaging the membrane. So now these cells for rest of their life lives a very different existence. They are only using these membrane proteins out there for their balance, osmotic balance, and some degree of active transport. But active transport is a very peculiarly controversial topic, but because where the energy is coming from, because you are not having a mitochondria out there. So that's why it's kind of pretty icky, but certain degree of transport, active transport happens. So, which essentially maintains its osmotic balance and thereby for rest of the life these cells. So, the structure what I showed you earlier. Now, this structure becomes almost, so these are once again these are individual cells which becomes like this. So, those cells now becomes like this. Almost it looks like that there is a sac filled with alpha crystalline proteins, and a very similar scenario happens in the case of hemoglobin, where in the case of RBCs, where a sac filled with hemoglobin. But now the fact is these are individual cells. You can keep on filling it. Okay. So, this is how the lens becomes a structure which is made up of cell with all the organelles loses it. This kind of programmed death has lot of connotation in physiology. Like say for example, if I have a way to target I always think over it, if I have a way to target the cancer cells, exactly like that, think of it, because cancer cells are very, very active in terms of their metabolism. They compete with the host. They pretty much deprive the host of nutrients. If there is a way that you can transform it, you target in such a way that those cells follow these kind of death. So, what will happen, even if the cancer cells are there, but they will eventually die off because they will not be able to really compete at such a faster pace as they do. I mean, that is where lies some of our futuristic medicine, like this is something which always strikes me. If a cell could be programmed, but then one has to be very careful it should not happen to some other cells, the live cells. You have to target it in a very, very unique way, that where you can transform some of these cancer cells into these kind of structures. Okay. So, this is just kind of a dreamy thought which always strikes me when for the first time I read this whole process of how the lenses are being formed. So, now what happens is something more to this. What is cataract? Now, that question I have an answer. So, now what happens, these cells, so you are having, now let us go to finer details of it. So, the finer details are this whole blue thing. Now, I am going to the finer details with, so these are nothing but alpha crystalline protein. Now, I am putting them in yellow. So, imagine these are individual molecules of alpha crystalline proteins, and it is very interesting to note that these alpha crystalline proteins which are filling this, which I am showing as the summation with the blue this thing, and now I am putting the yellow. So, these individual alpha crystalline protein, now I am showing in orange, has a particular pattern of folding, and that pattern is unique in terms of, so it has the N terminal, it has a C terminal. This alpha crystalline is unique in terms of allowing light to pass. But if this folding pattern, so there is a unique folding pattern of alpha crystalline, and theoretically speaking, this folding pattern is maintained throughout the life. And in order to maintain this folding pattern, this osmotic balance and all other properties are very essential. Now, say for example, this folding pattern, what you see a three-dimensional folding pattern in terms of alpha crystalline. Say for example, if I just outline the structure like this. So, this is the final folded structure I am getting. Say for example, for some stupid reason, something which we do not understand, this folding pattern changes. So, I will show you some changes which are happening. Say for example, it becomes like this, or some spot this becomes like this, or at some spot it becomes like this. So, then what happens is that its innate nature of allowing the light to pass is, this part is compromised. Okay. And once this part is compromised, so automatically what will happen? These yellow spots will now turn to a black spot. Initially there will be 1 or 2, but if it increases, then there will be more. This is the beginning of, if you remember, one of the picture I showed you, if I go, these black spots what you see, which are emerging out on the lens, are partly because of the misfolding of the alpha crystalline proteins. And that is where it requires, there are two possibilities how you can get around it. One possibility is you shine, that is what I was trying to tell you, you shine certain lasers out here, you burn that part. So, in other words, say for example, I know, so now let us look at the lens structure and then I will tell you. So, this is, say for example, a particular part becomes blackened, okay. Say for example, like this. What I do, I take a laser out here, which will, I shine the laser in this region. So, yellow is showing the laser, okay. I shine the laser and that kind of got rid of this. So, what will happen, the surrounding blues will try to migrate to that spot, they will migrate to that spot, the healthy ones, and with certain degree of adjustment, readjustment, one can restore the condition by virtue of light will pass through it. Okay. But that is a temporary solution. As aging will progress, this strategy will also falter. So, then you will be left with only one strategy, that you replace the whole lens with an artificial lens. So, that is the only other way by virtue of which you can get around it, and that is what is cataract surgery is all about. From the economic significance point of view, if we understand clearly, there are two forms of blindness. One is blindness which happens to the aperture, which is your cornea. The other one is the cataract. And India suffers from mostly these two blindness. But there is a third level of blindness, which is neuronal level, which is the damage to the damage out here, which is retinal blindness, or damage here, blindness due to damage to optic nerves. This is the another form of blindness. Then you have damage which happens, there is 3 level I told you, at the cortex, visual cortex, where your central processing unit, CPU, the real image recognition software where it is sitting, is damaged. Well, if the CPU gets damaged, I do not know what are our options, how we really can get around it. And to the best of my understanding, still no one is daring to hit on that problem. But for the retinal and for the optic nerves, there are possibilities. So, as I told you, most of the Indian population suffers from cataract or corneal blindness. But as most of you are aware of, or who sees TV and radios must have aware of, the India will soon become the diabetic capital of the world. A huge chunk of Indian population is suffering from diabetes. God forbid, we really do not know why it is happening, but it is creating a lot of issues in terms of retinal problems like diabetic retinopathy, because the blood vessels, as I showed you, these blood vessels which are present out here, if you look at it carefully, this is the zone where I am just now putting the red bordering. The blood vessels which are present there, because of diabetes, there is an increase in pressure in the blood vessels, and these are very thin capillaries. As we will go to the capillaries, you will realize it. They are very, very thin capillaries, and because of pressure on them, they burst, and once they burst, they kind of create damage on the retinal pigment epithelial cell as well as the retinal neurons, and those kind of damage are critical and will be critical in the years to come, while we will be talking about diabetic retinopathy and all other things. So, now after talking to you about these two, corneal and the lens, I will move on to the third level, which I am now putting a line, is retina. The structure of the retina, how it looks like and how it passes information, and what is dark current and what is the arrangement of these layers. So, let us close in here. In the next class, we will talk about the retina. Thank you.